So just to recap, on the outside of the eye, we have the cornea, the sclera. I removed all the adipose tissue and extraocular muscle. Let's see if I can find one specific extraocular muscle for you guys before I move on. That was the stuff that kind of looked like steak. And we saw the optic nerve at the posterior side of the eye. Now to view some internal structures, we are going to have to cut through the sclera. So imagine that the cornea or the front of the eye is the north pole and the optic nerve at the back of the eye is the south pole. If this is north pole and this is south pole, then the equator would be right around here. So we are going to make a cut just into the sclera right at this equator line. Not cutting too deep, definitely not cutting near the cornea. And so to get started, there's a couple ways you can do this. My scissors are pretty sharp, so I'm just gonna make a little snip if I can. And as you're seeing, it's not cooperating perfectly with me, but there we go. Just a small snip. I'm gonna try and keep in the straightest line possible and just cut straight through the sclera. Now you'll start noticing some fluid leaking out. When we get the eye opened, you're going to see that there is a ton of fluid inside our eyeballs. Most of the important stuff for vision is not in the middle of the eye, but is rather at the front of the eye or the back of the eye. So the fluid kind of just fills space and allows for light to pass through our eye the way it needs to. All right, I'm back at the beginning. Now we're gonna open up our eye. My students hate when I say this, but it makes sense, like an Easter egg. So there's a front and a back. And this is the internal anatomy of the cow's eye. Okay, so this is the front half of our eye, and we'll talk about what this little thing right here is in a bit. Let's focus on the back half of the eye. There is a tan, fleshy membrane back here. You can see I can kind of lift it. Normally it lifts off a bit easier, but this one's breaking on me. And this is known as your retina. And your retina is where you actually receive that light information and you translate it into electrical information that your brain can make sense of. So when light hits the retina, as it travels through the cornea, through the eye, and directly hits the retina, there are two types of cells in the retina that gather that light information, and we call these cells photoreceptors because they receive or they gather light. One type of photoreceptor is known as a rod cell, and rod cells work best when light levels are low. So rod cells are what you use in the middle of the night when you're trying to wander through your house if you need to take a bathroom break in the middle of the night and find your way to the bathroom. Now, when you're going through your house when it's pitch black at night, you can't see color, you can only see shades of gray or black and white. And that's because your actual rod cells back here in your cornea, which work in low light levels, they are not capable of color vision, and they're only capable of showing you that dark, shadowy vision. So the other type of cell inside your retina that are photoreceptors are called your cone cells. And when you think cone cells, think color. Cone cells are what you are probably using right now to watch this video if you're seeing it in color. Cone cells are capable of color vision, and they're the main types of photoreceptor cells that you use during the daytime. So our retina is filled with rod cells and cone cells, and the retina itself translates all that light information into electrical information that our brain can then make sense of through the optic nerve. Okay, let's talk about the elephant in the room. What is this goop? This goop is known as your vitreous humor, and the vitreous humor is a jelly-like substance that when the eye is all together, fills the entire eye and helps to maintain its shape. When the cow was alive, it was probably a bit more clear, so light could travel through it very easily. And in order to see the structures at the front of the eye, we're going to need to remove the vitreous humor. Now, there's no great way to do this. You can use your forceps to kind of scoop little pieces of the vitreous humor out, but it kind of gets frustrating because it likes to stick. You can also use your finger, and that usually tends to work a little bit more efficiently to scoop some of the vitreous humor out. And if you'd prefer at this point in the dissection, you can get a paper towel to get that vitreous humor out of there. There's going to be some lingering here. There we go, I got most of it. And now we can see a structure of the eye that you've probably heard of before. Right here, this is the lens. But before we talk about the lens, we've got to loop back here to the back of the eye. All right, I am going to remove the retina. Remember, that's this layer that's filled with rod and cone cells. And as I remove the retina, we are gonna see my personal favorite structure in the cow eye. By the way, the retina is really loosely attached. The only place it does attach is where the optic nerve is. If you've ever heard before that you have a blind spot in your vision, you really do. You can't tell right now because your eye compensates for it. But if you do a test, sometimes you can see where your blind spot is. That's right here because there's no rod or cone cells here where the retina attaches to the optic nerve. All right, do you see it yet? There's this vibrant bluish greenish structure. And we'll talk about that in just a moment. Unfortunately, we're gonna wait a bit. Hopefully your retina comes off a lot more easily than mine did because that was a disaster. But that's enough retina removed to actually see what we've got going on back here. So the first structure to point out is really the whole thing you're seeing right now. It just has a special bluish green part to it. This dark structure is known as the choroid coat. And the choroid coat is filled with blood vessels. There's not a lot of blood vessels on the inside of your eye, and there were really not any in the retina itself. So the choroid coat has all of these blood vessels, and it gathers nutrients and it gathers oxygen, and it feeds the retina those nutrients and those oxygen. So dark layer here is the choroid coat. But in the cow eye, a specific part of the choroid coat is this bluish greenish layer here. I'm sorry to disappoint, but you don't have this bluish greenish structure in the back of your eye. Your dog does, your cat does, a deer does, and a large number of other animals do, like horses, but humans do not. And the reason why we don't have this is if it's the middle of the night and I shine my flashlight in someone else's eyes, thank goodness their eyes don't shine back at mine. But if I'm letting my dog out to go to the bathroom in the middle of the night and I'm having trouble seeing her, and all of a sudden I'm shining my flashlight in her direction and she looks at me, she gets what we call eye shine. Eye shine happens only in animals that have what we call a tapetum lucidum. So a cow standing in a pasture in the middle of the night, if you shined a flashlight towards its eyes, that eye shine that you see is a result of the tapetum lucidum. Just a quick edit to this video for your pronunciation purposes. The structure that I'm referring to is actually pronounced the tapetum lucidum. So instead of pronouncing it in my Pittsburgh accent, tapetum lucidum, you can make sure you pronounce it correctly as tapetum lucidum. All right, back to the video. And this tapetum lucidum, it just makes these animals who tend to be out at nighttime a little bit better at seeing in the night. It takes that light that's sent to the back of the cornea and it reflects it back in the light so that now the eye just has more light to see with. So a lot of animals who have a tapetum lucidum are nocturnal, and that makes it a lot easier for them to see at nighttime. At the front of the eye, I want to talk about the lens. Now, the lens of the eye is just like the lens of a magnifying glass. Again, when the cow was alive, the lens was a lot more see-through and clear than it is right now. And if you are dissecting an eye that's not preserved but is fresh, you might even still be able to see through the lens. But what the lens does is similar to what maybe you've seen before when you have a magnifying glass on a sunny day. Whenever the light shines through the eye, the light goes through the cornea and then it goes through the lens. Now the lens acts like a magnifying glass on a sunny day, that it takes that light and it focuses right in the back of the eye at the retina so that all of the light is focused on the rod cells and on the cone cells, allowing your eye to see best. So just like a magnifying glass, the function of the lens is to focus the eye exactly where it needs to go on the retina. We are going to remove the lens, so you can do this by hand or you can do it with forceps. You just want to try and not remove any extra structures. And as I remove the lens, I may have popped it earlier, there is a little bit of fluid right where the lens was. If I poke here, right now I'm poking the cornea, so we've kind of gone the whole way through and you can see the cornea. It's this solid sheet, it's on the outside of your eye. This fluid that kind of gushed out here is called the aqueous humor, and it's just a water-based fluid, pretty much 100% water, that maintains the shape of the cornea so the front of the eye doesn't cave in. And here is the lens. The final structures I want to show you are ones that you are very familiar with. This space right here is the pupil of the cow eye. So that's like the black pupil of your eye. And it is black in a cow when all of the vitreous humor and all the other structures are inside the eye when you look into it, because all of that light that comes through the eye, it gets absorbed into the eye and it doesn't reflect back out. The eye just keeps all the light. So it looks black because the light does not reflect back. But this is our pupil. And then around the pupil, it's not a different color in this cow eye, but we have the iris. Now the iris is filled with tons of teeny tiny smooth muscles. So they're involuntary, they're out of your control. But you guys are familiar with what your pupil does. It's the job of the iris to contract those muscles, to dilate the pupil and make the pupil bigger in environments where there's low light. And it's the job of the iris to relax those muscles so the pupil gets smaller in environments where there is a high amount of light. So it is not the pupil itself that dilates itself and constricts itself in response to changing light levels. It's actually the iris that contracts and relaxes those muscles in response to changing light levels. And that is the dissection of a cow eye.